So welcome to the second video of the series, and this is about the Staphylococcus aureus lab diagnosis. So we know Staphylococcus aureus is a gram-positive cocci that are arranged in clusters. You can see this is a gram-positive cocci which are arranged in clusters when we see under the microscope. So first we know this order, like first we collect the sample. For the sample collection, we should know about the clinical features, like it can cause different clinical features and pathogenesis, it can cause suppurative infection, that is pus producing. So we collect the pus from these samples. In the respiratory infections like pneumonia, it can cause necrotizing pneumonia, etc. For those we can take the sputum. For urinary tract infections like pyelonephritis, we take the midstream urine. And for the food poisoning is caused by the enterotoxin produced by Staph aureus. So food poisoning, we generally get diarrhea, abdominal pain. For that we take the feces and vomitus. And for nasal, perianal and blood can also be collected, like blood we collect in like bacteremia, that is sepsis. Okay, this is about the sample collection. Next we have the direct microscopy. So under direct microscopy we have seen the gram-positive cocci arranging clusters, gram-positive cocci clusters, okay, grape-like clusters. Okay, next we have the culture. We know this order. Once you pause and see this order and try to remember without seeing. So first we come to the nutrient agar. So in nutrient agar, remember whenever we hear the word Staphylococci, we should know that they produce yellow colonies, yellow. Okay, so these colonies are circular in shape and it produces golden yellow color. Okay, you can see this is the nutrient agar plate, and we can see it is almost golden yellow color, and they are spherical colonies, and they are smooth convex. When we see from the side view, they are convex and they are opaque. So this is about the nutrient agar. This is the nutrient agar. Okay, next we have nutrient agar slope. In nutrient agar, golden yellow colonies are formed. So in nutrient agar slope also golden yellow and confluent growth is seen. Okay, you can see golden yellow colonies. And in the blood agar, it produces beta hemolysis. We know three types of hemolysis are there: alpha, beta and gamma. So it produces beta hemolysis in the blood agar. You can see this is the blood agar. Surrounding the colonies we have the beta hemolysis. Okay, for hemolysis you see other videos. I'm just talking about the lab diagnosis because you should all know what is hemolysis by this time. Okay, or you can see other videos, no problem. And in McConkey's agar, it forms a small pink colonies. So pink colonies means it is fermenting. Okay, lactose fermenting. So next we have okay, liquid media. It produces a uniform turbid growth in peptone media. Okay, in the selective media, selective media means the media which inhibits the other bacterial growths. So in that salt is added, because salt is not a problem for Staphylococci aureus, but it will inhibit the other bacteria, means it is halophilic. Staph aureus is halophilic. So we have three media, that is mannitol, salt, agar, and it produces again yellow colonies. And two media, that is salt, milk, agar and Ludham's media are also used. Their composition is written here: the nutrient agar plus 6.5% NaCl and 10% skimmed milk. It is about the salt milk agar. And Ludham's medium, lithium chloride and terulite is added to that medium. This is somewhat PG bits. So next we have the biochemical test. We have catalase positive, that is Staph aureus. Catalase negative is Streptococcus. You remember that. Staph and Strep differentiated by catalase test. Catalase positive, Staph, negative, Strep. And it shows fermentative type of in oxidative fermentation test. So it has a specific test called as coagulase test, and this is very important. They ask in many exams. So I'll tell you about coagulase test. This Staphylococcus aureus produce an enzyme called as coagulase. So this enzyme, what it does is, whenever it gets into our plasma, this coagulase will bind to a plasma protein that is called as CRF. Okay, CRF. This is called as coagulase reactive factor. So whenever coagulase binds to CRF, these both will activate prothrombin. So this prothrombin will be converted into thrombin, and it will activate fibrinogen to fibrin. So what will fibrin do is the formation of clot. Okay, this is the main coagulase test in the process of coagulation. Okay, so we have in the coagulase test, we have two types: tube coagulase